Hello students, in today's video we are going to study pharmacology of targeted cancer therapy. Now targeted therapy specifically targets and destroy the cancer cells with minimal effect on the normal cells. Now targeted therapies primarily include small molecule synthetic drugs and monoclonal antibodies. Now small molecule drugs enter inside the cells and they target proteins like tyrosine kinase. They can also target receptors like receptor tyrosine kinase that are overexpressed and autophosphorylated in cancer cells. So, by inhibiting tyrosine kinase and by inhibiting receptor tyrosine kinase, these drugs block signals that cause proliferation of cancer cells. Now, all small molecule drugs end with nib, like for example, imatinib, nilotinib. So, it ends with the nib. Erlotinib, it ends with the nib. Now, on the other hand, monoclonal antibodies act on the surface of cancer cells. They bind to specific antigens on the surface of the cancer cells so that these cancer cells are highlighted and then they are destroyed by the immune cells of the body. Now, monoclonal antibodies end with MAB. They end with MAB, like rituximab, cetuximab. So, this is about small molecules and monoclonal antibodies. Now, let's study the classification of targeted cancer therapy. Now, targeted cancer drugs are classified into 5 main categories. Now, the first category is of tyrosine protein kinase inhibitors. The drugs are imatinib, nilotinib. Now, these drugs inhibit tyrosine kinase. Then, a second category is of epidermal growth factor receptor inhibitors. Now, epidermal growth factor receptors are the receptor tyrosine kinase. So, these drugs, namely the gefitinib, then erlotinib, these are epidermal growth factor receptor inhibitors. Now, next category is angiogenesis inhibitors. The drug is sunitinib. Now, it prevents growth of new blood vessels in the cancerous tissue. Another category is the proteasome inhibitors. Now, proteasome inhibitors induce apoptosis or programmed cell death of cancerous cells. The drug is bortezomib. Now, next category of targeted drugs are the monoclonal antibodies. They specifically bind to the antigens on the surface of cancer cells and mediate their destruction. For example, rituximab, cetuximab, then bevacizumab. Now, before discussing pharmacology of each category of these targeted drugs, let's understand how tyrosine kinase and receptor tyrosine kinase induce cell proliferation and cause cancer. Now, tyrosine kinase is a protein or enzyme. Now, tyrosine kinase phosphorylates intracellular proteins and enzymes. So, look at this figure. This is tyrosine kinase. It has 2 binding sites. On one binding site, it binds to the substrate. Substrate is a protein or enzyme that is to be phosphorylated. On the other site, it binds to ATP. So, this is the ATP binding site. Now, this ATP or adenosine triphosphate has 3 phosphates. So, one phosphate from this adenosine triphosphate is transferred to the substrate and the substrate gets phosphorylated. So, tyrosine kinase phosphorylates and activates the substrate. Now, this activated substrate or this activated protein activates other enzymes. It activates other proteins. And thus tyrosine kinase initiates a cascade or series of phosphorylation, and thus one by one all proteins are activated and intracellular signals responsible for cell proliferation are produced. So this is the functioning of tyrosine kinase in a normal healthy cell of the body. So this tyrosine kinase is required for cell division, multiplication and proliferation. Now, very important to understand that tyrosine kinase is overactive in most of the cancer cells. Now, as we all know, cancer occurs due to the mutation in the genes or DNA. So, mutated cancerous genes produce specific overactive tyrosine kinase. Thus, overactivity or enhanced activity of tyrosine kinase causes excessive cell proliferation that results in cancer. Now, protein kinase inhibitors like imatinib binds to ATP binding site of tyrosine kinase. So, this prevents phosphorylation or activation of the proteins and enzymes. So, this stops series or cascade of protein phosphorylation, and thus downstream signaling pathway is blocked, and therefore proliferation of cells also stops. So, this is how the protein kinase inhibitors stops proliferation of cancer cells, and they control the cancer. Now, after tyrosine kinase, let's understand functioning of receptor tyrosine kinase. Now, receptor tyrosine kinase is the tyrosine kinase that is attached to a receptor. So, look at this figure. This is the receptor tyrosine kinase that is found attached to this receptor. Now, this receptor is a transmembrane receptor. It is found embedded in the plasma membrane. Now, this is the extracellular ligand binding site of the receptor. This is transmembrane helical peptide chain, and this is the intracellular site of the receptor which possess the enzyme tyrosine kinase. And since this tyrosine kinase is attached to the receptor, it is termed as receptor tyrosine kinase. Now, when the ligand epidermal growth factor EGF binds to this site, single or monomeric receptors, they move laterally and they form the dimers. So, as you can see here, this is the dimer. Now, dimerization activates tyrosine kinase activity of this intracellular domain, and they phosphorylate tyrosine residues on each other. So, here phosphorylation of tyrosine residue is represented by the letter P. So, both receptor tyrosine kinase are phosphorylated, and thus they are activated, and they further mediate series or cascade of protein phosphorylation, and thus provide signals for cell proliferation. So, this occurs in a normal healthy cell. Now, let's understand what happens in a cancer cell. Now, this receptor tyrosine kinase, it gets autophosphorylated in the cancerous cells. That means in the absence of the ligand, the tyrosine kinase gets phosphorylated in the cancerous cell. Moreover, in certain epithelial cancers, there is overexpression of epidermal growth factor receptor due to mutations. Thus, this epidermal growth factor receptor is a tyrosine kinase receptor. Now, if a protein kinase inhibitor, like for example gefitinib, it binds to the ATP binding site of tyrosine kinase, it will block this phosphorylation of tyrosine kinase, and further it will prevent the cascade of phosphorylation of the proteins. So, this will stop the cell proliferation. So, this is how overexpression or autophosphorylation of receptor tyrosine kinase causes excessive cell proliferation and results in cancer. So now let's discuss pharmacology of each category of targeted cancer therapy. Now, first category of targeted drugs are tyrosine protein kinase inhibitors. Now, the prototype drug is imatinib. Now, this drug is given orally. Now, imatinib targets BCR-ABL tyrosine kinase. Now, BCR-ABL is a mutated gene. Now, this mutated gene produces mutated BCR-ABL tyrosine kinase. Now, this abnormal tyrosine kinase is overexpressed or excessively produced in chronic myeloid leukemia. Now, imatinib enters inside the cell and block ATP binding site of tyrosine kinase. So, this blocks cascade of phosphorylation of proteins and downstream signaling. So, this prevents cell proliferation. And therefore, imatinib is the drug of choice in chronic myeloid leukemia. Now, nilotinib is an alternative drug in imatinib resistant or non-tolerant cases of chronic myeloid leukemia. Now, another target of imatinib is the platelet-derived growth factor receptor. Now, platelet-derived growth factor receptor is also a receptor tyrosine kinase. So, this receptor has tyrosine kinase on the intracellular domain. Now, imatinib binds to the ATP binding site of tyrosine kinase, and again it prevents the phosphorylation of proteins, thereby prevent the proliferation of cells. Now, this receptor is overexpressed in dermatofibrosarcoma protuberans. So, imatinib is very effective in dermatofibrosarcoma protuberans. Now, another target of imatinib is the stem cell receptors and c-KIT receptors. Now, stem cell receptor and c-KIT receptors are also receptor tyrosine kinase. These are autophosphorylated in gastrointestinal stromal tumor, which is a rare tumor. Now, imatinib is the drug of choice in metastatic c-KIT positive gastrointestinal stromal tumor, that is a GIST. Now, adverse effects of imatinib are abdominal pain, vomiting, fluid retention, which can also cause periorbital edema, that is the accumulation of fluid around the eyes causing puffy eyes, then pleural effusion, that is accumulation of fluid around the lungs, myalgia, that is muscle pain, liver damage, and congestive heart failure. Now, imatinib is well absorbed orally and it is metabolized in the liver by cytochrome P3A4 enzyme. Now, drugs that inhibit or induce cytochrome P3A4 enzyme produce potential drug interactions with imatinib. So, this is the pharmacology of imatinib. Now, the next category of targeted cancer drugs is the epidermal growth factor receptor, EGFR tyrosine kinase inhibitors. Now, this receptor, that is EGF receptor, is also a receptor tyrosine kinase. This receptor also possesses the enzyme tyrosine kinase on its intracellular site. Now, EGFR is also known as human epidermal growth factor receptor 1, that is HER1. It is a receptor tyrosine kinase as shown here. It possesses tyrosine kinase on its intracellular site. Now, in some cancers like in non-small cell lung cancer, this receptor, it gets autophosphorylated. That means even in the absence of ligand epidermal growth factor, this tyrosine kinase is phosphorylated, and because of which the signals are produced for the proliferation of cells. Now, the drugs like gefitinib, erlotinib, these are given orally, and they bind to the ATP binding site of tyrosine kinase, thereby they block phosphorylation of proteins and they block the signaling within the cell responsible for cell proliferation. So this is how they control the cancerous growth of the cells. So these drugs are indicated in non-small cell lung cancer, and erlotinib is also indicated in pancreatic cancer. Now, most common adverse effect of gefitinib is the skin rash and diarrhea. Other adverse effects are nausea, anorexia, itching, rise in serum transaminases. Then intestinal lung disease occurs infrequently and it is a serious complication. So this is the pharmacology of gefitinib. Now, another category of targeted cancer drugs are angiogenesis inhibitors. Now, cancer cells produce vascular endothelial growth factor. Now, this factor attaches itself to vascular endothelial growth factor receptors and stimulate growth of new blood vessels. Now, these new blood vessels provide nutrition to the solid tumor. Now, these receptors are tyrosine kinase receptors. As you can see here, the tyrosine kinase is found attached to the intracellular site of these receptors. Now, sunitinib is a vascular endothelial growth factor receptor tyrosine kinase inhibitor. So, sunitinib competitively blocks the ATP binding site of tyrosine kinase. So, this prevents phosphorylation of angiogenic proteins, and thus angiogenesis, that is the synthesis of new blood vessels, is prevented. Now, in the absence of blood supply, tumor cells degenerate. Now, sunitinib also inhibits platelet derived growth factor receptor alpha and beta, c-KIT, RET, etc. Now, sunitinib is used in metastatic renal cell carcinoma and resistant gastrointestinal stromal tumors. It is administered orally, and adverse effects are hypertension, rashes, diarrhea, weakness, bleeding, then proteinuria, hypothyroidism, neutropenia, and rarely it produces congestive heart failure. So, this is the pharmacology of sunitinib. Now, the next category of targeted cancer drugs are the proteasome inhibitors. Now, proteasomes are proteolytic enzymes that break protein. So, look at this figure. This shows a proteasome. Proteins are broken down to amino acids. Now, several signaling proteins control cell cycle and apoptosis. So, signaling proteins regulate programmed cell death of abnormal and mutated cells. Now, proteasomes, they degrade or they break these signaling proteins. So, this prevents apoptosis of mutated cells, and the cells proliferate, and this causes cancer. Now, in addition to this, proteasomes also regulate nuclear factor kappa B mediated signaling in the cells, which is responsible for inhibition of apoptosis of mutated or abnormal cells, resulting in proliferation of cells causing cancer. Now, this nuclear factor kappa B is a transcription factor. It is found in the cytoplasm, and it remains inactive because it is found attached to a inhibitory protein, that is inhibitory kappa B. Now, certain factors like hypoxia, that is lack of oxygen, cytotoxic drugs, degradation of DNA, and other stressful stimuli activate this proteasome. So, proteasome breaks inhibitory kappa B that releases nuclear factor kappa B. Now, this nuclear factor kappa B inhibits apoptosis of abnormal cells, and that results in the cell proliferation, and that further causes cancer. Now, this nuclear factor kappa B is overexpressed in some cancers. Now, proteasome inhibitors will inhibit this proteasome, and they will prevent all the activities of proteasome. So, nuclear factor kappa B will not be released. So, mutated abnormal cells, they will undergo apoptosis. So, this will stop the cell proliferation, and this will further stop the growth of cancerous cells. Now, bortezomib is given IV. It is a proteasome inhibitor. It covalently binds to proteasome and inhibits its proteolytic activity. So now inhibitory kappa B is not destroyed, and since inhibitory kappa B is not destroyed or it is not degraded, nuclear factor kappa B is not released, and therefore abnormal cells, they undergo apoptosis. Abnormal cells, they undergo programmed cell death, and therefore the cells, they do not proliferate. Now in addition to this, since the protein breakdown is prevented, this causes accumulation of Bex protein in the cells, and this Bex protein promotes apoptosis of abnormal and mutated cell. So overall, this stops the proliferation of cancerous cells. Now, bortezomib is used along with other drugs as a first-line therapy for multiple myeloma, and it is also used for refractory mantle cell lymphoma. Most prominent adverse effect is peripheral neuropathy. Other adverse effects are diarrhea, fatigue, bone marrow depression, especially thrombocytopenia. So, this is the pharmacology of bortezomib. Now, another very important category of targeted anti-cancer therapy is the monoclonal antibodies. Now, antibodies, as we know, are the proteins that recognize and destroy the antigens. Now, antigens are foreign substances which are found in the body like toxins, bacteria, viruses, etc. Now, these monoclonal antibodies, they destroy the cancer cells. Now, monoclonal antibodies are identical antibodies that are synthesized in the laboratory and are active against specific antigens. So, look at this figure. Here, this green color, rituximab. Rituximab is a monoclonal antibody and it binds to CD20. Now, here CD20 are the specific unique antigens which are found on the surface of the malignant or cancerous B cell. Now, binding of monoclonal antibody rituximab to CD20 highlight this malignant B cell, and therefore this malignant or cancerous B cell is easily identified by the immune cells of the body and are killed primarily by three methods: apoptosis, then antibody dependent cellular toxicity, that is ADCC, and complement dependent cytotoxicity, that is CDC. Now, binding of monoclonal antibody, that is rituximab, to CD20 triggers apoptosis. That means it triggers the programmed cell death of this cancerous B cell. Now, monoclonal antibody also activate effector cell, that is effector T and B cell of immune system, that kills the target malignant cell. Now, in addition to this, binding of rituximab to surface antigen on the cancer cell also activate the complement pathway. So, multiple serum proteins or multiple complements are activated that kill the target cancer cell. So, this is the mechanism by which monoclonal antibodies kill the malignant or the cancerous cells in the body. Now, let's discuss pharmacology of some monoclonal antibodies used in targeted cancer therapy. Now, all monoclonal antibodies are given by IV infusion, and these can be either chimeric or humanized. Now, name of all the monoclonal antibodies end with mab, like rituximab, cetuximab. Now, first monoclonal antibody we are discussing is the rituximab. It is a chimeric monoclonal antibody. It binds to CD20 present on the surface of malignant B cells. We have already discussed mechanism of action of rituximab, how rituximab destroys the or kills the malignant B cells. Now, rituximab is indicated in B cell lymphoma, non-Hodgkin lymphoma and chronic lymphocytic leukemia. Now, administration of monoclonal antibodies can cause infusion reaction. which is characterized by chills, fever, urticarial rashes, pruritus, dyspnea, that is difficulty in breathing, and hypotension. Now, severity of infusion reaction varies and is also related to the rate of infusion. Now, as rituximab attacks the B cell, depletion of B lymphocytes and neutropenia can also occur. So, this is the pharmacology of rituximab. Another drug is the cetuximab. Now, cetuximab is a chimeric monoclonal antibody. Now, it targets EGF, that is the epidermal growth factor receptors. Now, these receptors are receptor tyrosine kinase. They possess tyrosine kinase on the intracellular site. Now, in place of EGF, cetuximab binds to the ligand binding site of these receptors. So, this prevents phosphorylation of tyrosine residue, and further it prevents phosphorylation of signaling proteins. So, this prevents proliferation of cells. Now, cetuximab is indicated in squamous carcinoma of head and neck and colorectal cancer. Adverse effects include infusion reaction. Now, anaphylactoid reaction may also occur during the IV infusion. Then hypomagnesemia and interstitial lung disease can also occur, but this is very infrequent. So, this is the pharmacology of cetuximab. Now, another drug is bevacizumab. Now, it is a humanized monoclonal antibody. Now, this drug, this monoclonal antibody, bevacizumab, it binds to a growth factor. It binds to vascular endothelial growth factor. Now, because it binds to this growth factor, it prevents the binding of vascular endothelial growth factor to this receptor. Now, this receptor is also a tyrosine kinase receptor. So, inhibition of or prevention of binding of this growth factor to this vascular endothelial growth factor receptor prevents the phosphorylation of tyrosine kinase, and that further prevents the phosphorylation of other angiogenic proteins. So, angiogenic proteins are not activated. So, angiogenic signals are not produced, and therefore, the growth of new blood vessels stops. So, this prevents growth of new blood vessels in the cancerous tissue, and in the absence of blood supply, the cancerous tissue degenerate. So, that is why it is termed as a angiogenic inhibitor also. Now, bevacizumab is indicated in colorectal cancer. It is indicated in non-small cell lung cancer, then breast cancer, renal carcinoma, and glioblastoma. Now, administration of bevacizumab can trigger infusion reaction, and other adverse effects are rise in the blood pressure, then arterial thromboembolism, that is the blockage of arteries by a thrombus or by an emboli, and this can cause heart attack and strokes. Now, besides these, blood vessel injury, then hemorrhages, heart failure, proteinuria, gastrointestinal perforation, and healing defects can also occur. So, this is in brief on pharmacology of monoclonal antibodies and small molecule synthetic drugs used in targeted cancer therapy. Now, please note, information provided in this video is only for academic informative purpose. For use of any of the targeted drugs or for the treatment of cancer, consult your physician. If you find the video useful, kindly like, subscribe and share this video. Thanks for watching this video.